Our next speaker is Ricardo Feldman, who will talk about acid ceramidase plays a key role in the pathogenic cascade leading to neurodegeneration in Gaucher and GBA-associated Parkinson's disease. Let me just find your slides.
Thank you for inviting me to this very exciting meeting. I'm really honored to be here. So in our lab, we use induced pluripotent stem cells for modeling Gaucher and more recently GBA1-associated Parkinson's disease. And using this system, we found that there are problems with the lysosomes. There is lysosomal dysfunction, like it was known. But we also found that there were developmental defects in the hematopoietic system that recapitulate the anemias. There was defective osteoblast differentiation, which recapitulates skeletal abnormalities. And there was also impaired neuronal development. So this is work that has been published, but is to frame my talk. There is an elevation of glucosylsphingosine in neuropathic Gaucher disease in several samples, GD2 and GD3. And what we found was that when we made neuronal cells from neuropathic Gaucher disease, there were elevated levels of glucosylsphingosine that caused hyperactivation of mTOR, and that led to lysosomal dysfunction and autophagy defects. And then when we use an inhibitor of acid ceramidase, carmofur, then we lower the level of glucosylsphingosine, mTOR was not hyperactivated, and we had lysosomal rescue. And of course, the lipid came down. So the question here is, does acid ceramidase also play a role in GBA-associated Parkinson's disease? Now, this is a complex question, and there are a few facts that we have to consider. Only a small percentage of GD patients develop Parkinson's disease, and there is a similar low pH Parkinson's frequency in GD carriers, even with severe mutations. I apologize for using the name severe here. Due to low penetration of the GBA mutations, there are other factors that become important, like genetic background, age, environmental, and other epigenetic factors, and perhaps even others that we haven't thought about. So to study the contribution of mutant GBA to PD, we need a very stringent genetic system, and for that we resorted to isogenic controls. We received iPSC from Michelle Adelaide, and the gene edited controls. So we use N370S, L44P, REC-NCI, and the corresponding wild-type controls. Here at the right, we show that the activity of GCase, the enzyme, was lower in the mutant, but it was higher when it was restored when we use the gene-edited lines. This is what the cells look like, stained with tyrosine hydroxylase. These are dopamine neurons. And so we wanted to study this and look in detail at acid ceramidase, the effect of acid ceramidase on alpha-synuclein. And we did an experiment that was published. We looked at total alpha-synuclein, and there is an elevation in the mutant compared to the gene-edited controls in both the high molecular weight oligomers and phosphoalpha-synuclein 129, and this is quantitated here. And then when we use an acid ceramidase inhibitor, carmofur, there was a restoration, at least a partial restoration of the phenotype. And this is true for the high molecular weight alpha-synuclein and also for the phosphorylated alpha-synuclein. So we wanted to look at this more seriously. We know that inhibitors have off-target effects. So we use an additional 3 different inhibitors. These are the formulas, and they are all commercially available. And I will go, these are our 3 lines, the REC-NCI line, N44P, and N370S. But I will focus, we found the same results in all 3 lines with this particular inhibitor, the second inhibitor we used. And we can see that when we use this inhibitor, we can lower the level of the alpha-synuclein oligomer. And as I said, the same thing was true for the other two mutants. When we look at the second inhibitor, this one crosses the blood-brain barrier. Same thing. We can lower the level of high molecular weight alpha-synuclein and alpha-synuclein 129, and this is true also for the other two lines. And then same thing was true for 3 lines with the third inhibitor. So we wanted also to look for evidence, genetic evidence that this was true, that if we knock down acid ceramidase, we also lower the formation of the alpha-synuclein 129, and with the CRISPR-Cas9 knockdown, and we reduce it to 50%. This is equivalent to a haploinsufficiency, and we can see that the effect of this knockdown on the high molecular weight and the low molecular weight alpha-synuclein was similar. This mutant REC-NCI wild type had lower levels of alpha-synuclein aggregation when we did the knockdown, and I will not show you, because of time, the results with L44P and N370S, but we got similar results. And then we looked at markers of lysosomal function. We looked at this P62 marker, which was elevated in the mutant compared to the gene-edited control, and then when we looked at the knockdown, there was a substantial reduction, and the same thing for cathepsin D, which is lower in the mutant compared to the control, and then we were able to at least partially restore the level of cathepsin D. So we already heard about TFEB, which is a master regulator of lysosomal and autophagy genes, and that it is necessary for this transcription factor to go to the nucleus in order to exert its functions. So what happens is that in our hands, mutant GCAs will cause hyperactivation of mTORC1, and we heard about this from a beautiful talk from Greg. And when this TFEB needs to be dephosphorylated so it can go to the nucleus, but mutant GCAs interferes with this, and then there will be an autophagy lysosomal function block. So I want to point your attention to the lower panel, the purple. It shows that what happens when we use 3 different inhibitors of acid ceramidase with this mutant, but the same is true for the other 2 mutants. We see that in the gene-corrected lines, there is a lot of TFEB in the nucleus where it is functioning, and then there is very little in the mutant. And then when we use 3 different inhibitors, we see a restoration of the nuclear localization of TFEB, and this is quantitated here. And then CRISPR-Cas9 knockdown of acid ceramidase also rescue nuclear localization of TFEB. Again, I want to point your attention to the lower here, to the purple. The control cells have a lot of TFEB in the nucleus, and this mutant has much less in the nucleus, and when we have the knockdown, we have a restoration, a rescue of the phenotype. So to summarize, I think that we would like to think that we have validated acid ceramidase as a potential therapeutic target, and inhibition of acid ceramidase by 4 different small molecules decrease alpha-synuclein levels and aggregation. CRISPR-Cas9 knockdown of ASAH1 also decrease alpha-synuclein levels and aggregation. Both acid ceramidase inhibition, biochemical, pharmacological inhibition, and also genetic knockdown of ASAH1 rescue ALP functions involving alpha-synuclein clearance. That is, there is improved autophagy, there is an increased transfer of TFEB to the nucleus, and there is higher expression of cathepsin D. And as I said, I would like to propose that we consider acid ceramidase as a potential target for GBA-associated Parkinson's disease. These are the people who did the work. Manoj Kumar, who is a fellow of the Silverstein Foundation, did all the work on Parkinson's, and earlier a graduate student in the lab, Mansa Srikant, did all the work on Gaucher, and I want to thank our funders for this work, and thank you for your attention.
The oleoethanolamide that we discussed a while back in terms of acid ceramidase inhibition. I was wondering whether. Oh, sorry. I was wondering if you had any comparative data comparing the commercially available.
I don't think we did that.
Oh, you didn't end up doing it?
I
Because
don't
it's an
remember.
endogenous cannabinoid
Sure.
that also purportedly has acid ceramidase inhibition. I was wondering whether it's, like a homeostatic regulator, and whether you had a chance to look at
Yeah,
that.
I owe you that.
Oh, okay, fine. And the second one is the inhibitors. You said it lowers ACIN aggregation.
Yeah.
But what does it do to monomeric ACIN?
Also, it lowered the monomeric phosphorylated. I can go back and show you. So when we look at the inhibitors, there is more aggregated, but there is also more monomer here. These are triplicates.
Okay.
And then when we use the inhibitor, and this is true for 3 different inhibitors in 3 different cell lines, we lower not only the aggregate, but also the monomer.
But wouldn't that cause a loss of function of ACIN's normal signaling activity?
It's not that we eliminate it. We bring it down to control levels.
Ah,
the
okay.
same control levels that we find in the gene-edited line.
Okay, thanks,
Okay.
thanks.
Any other question? Go to the microphone.
Were you able to see these results also on IF, similar to how you presented later in the talk, where you see a reduction in phosphosynuclein in IF?
I don't think I heard the first part of the question. So
I'm just asking if this result on a Western, you could also see visibly on IF when you treat with the inhibitor versus not.
we did that, but I don't have the data here with me. Yes.
Okay. And how do you explain because then which of these species basically do you think is being targeted? Because you have multiple bands of phosphosynuclein that are higher molecular weight, which presumably are just different aggregate structures here. So
They are different aggregate structures. How this is targeted and how these different molecular weights are selected, I have no idea. I don't know. Sorry.
Thank you very much.